Hi, this is Miss Litton, and this is Chapter 36, Body Fluid Regulation and Excretory Systems. And this is intended for my AP Biology class. This is your EdPuzzle portion of this lecture. So we're going to be focusing on, in this first section, on animal excretory systems. And the big idea here is that most organisms have some sort of tubular organs to regulate saltwater balance and excrete metabolic waste. But of course, as always, we're going to start small and build our way up. And then we will discuss the human excretory system in the nephron in class. All right, so the big idea here on water balance is however much water comes in, that's how much water goes out. And you don't want to lose more than what you have. And your first term, osmoregulation, the definition for your notes, osmoregulation, balancing levels of water and salts in the body. Balancing levels of water and salts in the body. And usually we need to get rid of, well, we have to get rid of metabolic waste, and any process where we're doing that, that is referred to as excretion. Okay, so let's focus on the big one here, and that is nitrogenous waste. And the first thing I want to see if you can do this, if you remember the structure of an amino acid. And if you're at home watching this video, either imagine it in your head or actually commit it to a piece of paper, the structure of an amino acid. If you want to, you can pause and keep going. But this is what an amino acid does look like. You have a hydrogen group, you have a carboxyl group, an amino group, and an R group. And if you remember, how many different R groups do we have? Hopefully you said 20 different R groups. That's why we have 20 different amino acids. Also, this hydrogen oftentimes comes right on over here. So this ends up being NH3, a little plus here, and then this is O- over here. So the part we're focusing on for this part of our discussion is this, of an amino acid, this NH3. This is the nitrogenous waste that needs to get managed. We can also get nitrogenous waste from DNA as well, and we'll talk about that. The importance of amino acids, if you go in your notes on your introduction, we talked about how amino acids can be used to synthesize other proteins. If they are unused, they'll be oxidized for energy or stored as carbs or fats. And it's the breakdown of nucleic acids and amino acids that results in these nitrogenous wastes. And depending on the species, will depend on what we're going to do with this nitrogenous waste. So taking a look here at this diagram, up at the very top, you can see proteins, and you know proteins are built out of amino acids. And here we have that amino group off to the left. That's what we're looking at right here. So what are you going to do with it? Well, three options. One, you could add a hydrogen, already has it on there, and you'll have ammonia. But when you think about ammonia in your mind, hopefully you think of it as a toxic substance. This wouldn't work in our bodies, but for organisms with copious amounts of water available to them, it works just fine. They don't have to use much energy. If you look at the bottom, the red line is the energy needed to produce. So they don't need a lot of energy, but water, you need a lot of water, okay, to get rid of ammonia, to flush it out of your system. If you want to take this ammonia, you can convert it to urea. Now, urea, if you recognize here, you can see an amino group here on either side. And then actually what you do is you take a couple of those amino groups and you combine it with carbon dioxide, and that's how you form energy. But that is going to involve a reaction and input of energy. So if we look on our energy scale, we're moving up, but you don't need as much water to get rid of urea because it's not as toxic. Level it up one more time and you'll make uric acid. Uric acid, you can see it's a larger structure molecularly, but it does not require any water. If you look at your water level, very low. So the least amount of water, of all in order to handle uric acid, it takes a lot of energy to make it. But now if you're something like a bird developing within a shell and you have to stay there your entire development, then if you were generating ammonia, there wouldn't be room for your body to develop and all the water you would need to handle that ammonia. If you're in that shell and you're making uric acid with your nitrogenous waste, then you can store that as a solid, as a precipitate. So if you take a look at this diagram, it's talking about that. So where do our amino groups come from? They could come from our proteins, specifically amino acids, or from your nucleic acids. Either way, you have this amino group. So the different strategies, most aquatic fish, most aquatic organisms and many fish, what they'll do is they'll do the ammonia route because they'll have a lot of water available to them. Mammals like ourselves will take the extra energy step to convert it to urea. So it requires, we're terrestrial organisms, we're on land, we don't want to spend as much energy, we don't want to spend as much water. So we'll take the energy and convert it to urea. And then birds and insects, they'll expend even more energy and convert it into uric acid, which requires no water. And here's a little chart to explain that as well. And let's fill in our notes. So on our notes underneath nitrogenous waste products, ammonia is formed, and then, depending on the organism, little letter A, ammonia excretion. It is toxic, but it is water soluble. Little energy, but requires a great deal of water. Most fish and aquatic animals. Toxic, but water soluble. Little energy, but requires a great deal of water. Most fish and aquatic animals. B, urea excretion. Less toxic, so it conserves water. But it requires more energy to produce. Than ammonia. So examples of that, urea excretion are amphibians, adult amphibians, sharks, and mammals. Now adult amphibians, because juvenile, they're in the water and they have that available to them, so they can go the ammonia route. Okay, uric acid is least toxic. It requires the least amount of water, but it costs the most energy to produce. And insects, birds, and reptiles respond in this way. Okay, so you're going to have an EdPuzzle question inserted here, and this is what it is. So if you need to, you'll stop, or EdPuzzle will stop it, and then you'll be able to answer this question. All right, now, those are our nitrogenous waste products. So let's take a look at what happens with some invertebrates. Again, we're going to see the strategy of using tubes, as I mentioned at the beginning. And so the first one we'll look at is the Planaria. And we have already talked about the Planaria with our other systems. We're going to revisit it here as well. Within their branching gastrovascular cavity, you also have some excretory tubules. And these excretory tubules have cilia right here. These cilia will beat in a way to move the water along throughout their bodies. And so that's why it's referred to as a flame, because it's moving, kind of undulating like a flame would. And so for that one, under flame cells and Planaria, branching excretory tubules that open to the outside of the body through excretory pores, and fluids propelled through tubules by beating cilia that looks like a flame. All right, and now let's move on to another example. Let's look at the earthworm, where you have flame cells in the Planaria. In the earthworm, what you have is nephridia. And each segment has its own nephridia. And what you'll see on either side. So you'll have, for the first time, what you're seeing here are two tubes passing one another. One is part of their closed circulatory system, and one is for waste. And they're side by side. And hopefully you can see some advantages to that, because any waste that you have can move into the excretory tubes, and items that you want to recover, possibly water, can go back into the circulatory system because they're traveling side by side. Whatever you don't want ends up going here as this bladder and then out the nephridia pores. So on your notes, what I want you to have is nephridia and earthworms. Each body segment contains a pair of nephridium consisting of a coiled tubule surrounded by a capillary network. A coiled tubule surrounded by a capillary network. Nutrients are reabsorbed by capillaries and waste move into the bladder and exit through pores. Now, if you want to know how any fluids get into nephridia at all, there are, let me get a different picture here for you. Nephridiostomes. This portion right here extends into the segment in front of it. Fluid moves in and then curls around through here, and then you can see the purple is representing the circulatory system around it. So you're going to recover the nutrients back into the blood. Whatever you don't want goes in the bladder and out the nephridia pore. So the opening, it's kind of like a vacuum in the chamber in front of it. All right, and then the next example we want to look at are the Malpighian tubules that you find in insects. Now, you know, we've talked about this before, that insects have an open circulatory system. And you know that their gases are moving due to their tracheal tubes going through their open spiracles. But in their hemolymph, this is how they move their nutrients and their waste. So these Malpighian tubules extend out from their gut out into this hemolymph. And once again, it's going to be picking up waste, but also water from their hemolymph, and then moving through, they're calling this the hind gut and intestines, basic, kind of like a precursor to that, and then you have a rectum. Anything that the body wants back, like water or any nutrients, can get reabsorbed out, and then whatever is left over it exits the body. And keep in mind that insects take the energy to convert their waste into uric acid, so it can be a very dry waste. So for Malpighian tubules in insects, long radiating microtubules surrounding the gut that take up waste and water from the hemolymph. At the rectum, useful substances are reabsorbed and uric acid passes out the anus. All right, so that was just excretory organs among invertebrates. And now we're going to move to aquatic vertebrates. So here are issues. Do you live in freshwater situation or do you live in a saltwater situation? And words that should be coming to your mind right now are hypertonic, hypotonic, and isotonic. And you know water must flow from the hypo, and it's going to move into the hypertonic situation. So if you are an organism living in salt water, chances are that salt water, right, is going to be hypertonic to your body. So the environment is high in salt, and you're going to tend to lose water because you're going to be the hypotonic organism, and water is going to want to flow out of you and into that salt water, and salts are going to want to flow in. If you're a freshwater, this is a reverse. If you're living in fresh water, you're assuming it's unpolluted, etc. But if you're in freshwater, then your body is probably hypertonic, and water is going to want to flow into you. So you have a different battle depending on where you live. So let's look at some strategies to take care of this. Marine invertebrates and cartilaginous fish. So here's some marine invertebrates and cartilaginous fish like sharks. The way they solve it is they keep a high level of urea in their blood, and their cells can withstand that high level of urea in their blood, unlike us. And by doing that, they're making their body fluids isotonic to their surrounding medium, so they don't have to worry about water loss. So body fluids are isotonic, I meant cartilaginous fish. Body fluids are isotonic to seawater due to high urea concentration kept in the blood. Due to high urea concentration kept in the blood. All right, now let's look at bony fish. And we're going to start off with marine fish. And in this diagram, you can see the descriptions. All right, so keep in mind, if you're a marine fish, the fluid out here is probably hypertonic and your body is hypotonic. And you know water must flow from the hypo, so you're going to tend to lose water. Well, you're only going to lose it where it comes in contact with the environment, and you know one of these places is right underneath this operculum because this is where you have your gills. And if you recall, right, the gills, their job is you're trying to pick up oxygen and get rid of CO2 using the water. A word should come to your mind right now. It starts with a C. Hopefully you're thinking countercurrent, that the blood of the fish in its lamellae, the disc-like lamellae, are flowing countercurrent to the water to facilitate the transfer of oxygen from the water and into the blood. But it's those areas of the gills where they're vulnerable, because if their body is hypotonic here, then they're going to tend to lose water. Just passively, you're going to lose water through those gills. Now, when they're going to drink seawater to recover some of that water, so they're bringing it in through their mouth, and with that seawater is going to come the salt that is in their environment. So they have to actively transport salt at their gills. So they're passively losing water there, and they're actively transporting salt out of their bodies. Now, if you are tending to lose water to your environment, are you going to have copious amounts of urine or just a little bit of urine? And hopefully it makes sense to you that you would have scanty. That means a little bit amount of isotonic urine. You can't afford to pee a lot because when you pee, you are losing water. So on your bony fish, for marine, in your notes, prone to water loss, because they are hypotonic to the surrounding environment. So what do? They drink water. At the gills, they actively excrete salts that came in with the water drinking, and they have passive water loss. And then number 3, scanty isotonic urine. You're going to have the opposite problem for your freshwater fish. Hopefully these things make sense. If you're a freshwater fish, now your environment is hypotonic, okay, and your body is hypertonic. So water must flow from the hypo, so you're going to have an issue of too much water, and you're going to want to get rid of that excess water, so that's why you have large amounts of hypotonic urine. And you're going to be concerned about losing salt, because salt's going to want to go from a higher concentration in your body to a lower concentration in the water, so you have to actively take up salt by the gills because you will tend to lose it to your environment. And you don't need to drink because you have plenty of water. So you can see right here, passive gain of water across body surfaces and through gills. So for freshwater, prone to water gain because they are hypertonic to the surrounding environment, does not drink water. At gills, actively uptake salts and passive gain of water. And they have large amounts of hypotonic urine. So you can see now why you can't take a saltwater fish and put it in a freshwater tank and vice versa, except if you had some salmon. Salmon are really good osmoregulators, and they can move from freshwater or saltwater into freshwater and survive and not have a problem moving back into the freshwater where they originated. So when they have their net, so let me jump back here. I want to finish this first. I skipped over a slide. If you take a look here, this is a good diagram where it kind of puts it together for both freshwater and seawater. And you might want to grab this out of the PDF and add that into your group shared notes. Okay, and then on C, the salmon, excellent osmoregulators. They can alter both their gill and kidney functions alter both their gill and kidney functions, moving between fresh water and salt water. All right, here's some questions. I'll be asking you these on EdPuzzle. So here's your first one. Which of these is mismatched? Here's another question. One advantage of urea excretion over uric acid excretion is that urea. So how is urea excretion better than uric acid excretion? And your next one, freshwater bony fish maintain water balance by, you want to choose the right answer. Okay, and then this brings us to our last section, osmoregulation by terrestrial vertebrates. So we're on land, we're always going to tend to lose water, and you got to think about areas where we can lose water. So we have major adaptation of water conserving kidney. Major adaptation of a water conserving kidney. Again, your water in needs to equal your water out. What are ways that water comes in? Through the food we eat or through cellular metabolism. Remember, who catches the electrons at the bottom, oxygen forming water. So from metabolism. Also, any dehydration synthesis is going to form water. So ingestion and metabolism. How do we lose water? We lose water by having it evaporate from our skin and lungs, through our feces, and through excretion. So again, even though we're multicellular, always think of it as being just a conglomerate of single cells, and everything's going to go across our cell membranes. And when we think about body fluid regulation, our cells are surrounded by tissue fluid. And then our tissue fluid, because we are terrestrial vertebrates, we're going to have a closed circulatory system. Our blood passageways are going to pass through our tissue fluid, and that's where all of our exchange is going to take place. So here are some of the different ways that water could move. If you look here through our gut, through our lungs, when you exhale, every time you exhale, you're exhaling some of that moisture. Our kidneys play a factor in our water and ion balance, the biggest one, and we'll be talking about that in class. Our skin, we lose water, and urea and bio. And we'll break those down for us when we get into the lecture and class. Okay, so a couple of examples of osmoregulation by terrestrial vertebrates. One is the kangaroo rat. He lives in dry, desert-like conditions. He has very concentrated urine to conserve water, and he never ever drinks. And the reason is he is so good at conserving his water, and he gets all of his water from just the metabolism of the food that he eats. So on your notes, kangaroo rat, concentrated urine, dry feces. Concentrated urine, dry feces, gets all water from aerobic respiration. Some marine mammals and seabirds, they have a problem with too much salt, so they have salt glands to help them get rid of some of their excess salt. So on your notes, specialized glands to excrete salts. Several skin adaptations, thick, scaly skin, impermeable to water in order to protect themselves from water loss. And tardigrades, these are known as water bears. And something interesting, these are small microscopic animals. And some, they have been frozen for more than 3 decades, 30 years, and they were able to bring them back to life. And so they study them to look at maybe protecting our own tissues or discuss, how life might originate on other planets. Anyway, the process here is they have the ability to go from an active to an inactive state just through dehydration. And I gave you that example there. So these are nematodes, these are worms, so clearly these aren't vertebrates, but I wanted to just throw that in there. Plus they look cool. And then camels. Camels and kangaroo rats have convoluted nasal passages that absorb moisture from exhaled air. So when the air exits, it's not a straight tube like our nasal passageways. It kind of cycles around in order to get out, and it has multiple opportunities for that water to be recovered. All right, and let's see, I think that is going to be it. Yep, for you. So hopefully you answered all your questions correctly. Bye.